Welcome back to M.K.G BIOTECH CLUB. So today's topic is terminologies used in genetics. You know what is genetic? Genetics is the study of heredity and variation. So first we have to understand what is heredity and what is variation. Heredity is the phenomena by which living organisms transmit parental characteristics from one generation to the next generation. That means those characters which are transmitted from one generation to next generation are considered as hereditary characters, like the color of the eyes, hair color. So these are some characteristics which are very commonly observed between parents and offsprings. Although there are some variations as well, so the differences in characteristics between the offspring and their parents and with each other is called the variation. Variation may be morphological, anatomical, physiological, or behavioral differences between them. So I think both the terms are very clear to all of you, like heredity and variation. So don't confuse between these two terms. Another important terms are alleles and genes. So before going to discuss about the allele, I think first we have to take a look on genes. Gene is the fundamental unit of heredity, which is located in DNA in the chromosome. Actually, genes are transmitted from one generation to the next generation, which code for different in living organisms. Gene can form their exact copy during the replication of the DNA. So genes are the factors according to the Mendel which transmit one character from one generation to the next generation. So what are alleles or allelomorphs? Basically, an allele is one or more alternative form of a gene. So it has a specific position on a particular chromosome and code for a particular characteristic like the height, color of skin, color of eye, color of a flower, seed color. So these are some characteristics which are showing alleles. They code for different alleles. And if we talk about the height of a pea plant, so for dominant allele, capital T is used, and for recessive allele, small t is used. The very important terms are homozygous and heterozygous, and these are really very common terms in which students get confused because these are very commonly used in Mendel's law, in law of segregation as well as in law of independent assortment. Homozygous, homo word means similar or identical. So the organism having two identical alleles like capital T or small t. So these are identical pair of alleles of a gene coding for a particular characteristics. So these are considered as homozygous. While the non-identical genes, you can say non-identical alleles of a genes are considered as heterozygous. So like capital T and a small t when come together. So this is the condition of heterozygous. Like capital R and small r came together. So this is also a condition of heterozygous. Generally, dominant alleles are represented with the capital letter and recessive alleles are represented with the small letters. Next very important terms are phenotype and genotype. Phenotype are basically external appearance, or you can say those characters which can easily be observed by our eye. Like we can observe the color of eye, the height of any person or any plant, color of flowers. So these are some external appearance. So the external appearance of an organism is called its phenotype. It is expressed in words like tall, dwarf, red, white, etc. Parental characteristics of an organism cannot be known or determined by the phenotype of that organism. What does this mean? We want to explain that if we know that someone is tall, so we don't know whether it's that person is homozygous tall or heterozygous tall. Okay, just by seeing that person. But genotype is the genetic makeup or the genetic composition of the organism. It indicates about whether that organism is homozygous tall or heterozygous tall. Like capital T, that means pure tall or homozygous tall. Capital small t, that means hybrid tall. And small t, that means the organism is recessive or dwarf, you can say. Terms are dominant and recessive characteristics. Dominant are those characteristics which express themselves in all the conditions, whether they are present in homozygous condition or in heterozygous condition. If capital T is present, so it doesn't depend that the other pair is capital T or small t, the plant is tall in appearance. Okay, but recessive are those characters which express themselves only in homozygous condition. Like if we talk about the small t, if a small t present with the capital T, the resultant is a tall plant. So the small t hides its characteristics in presence of capital T. But if small t is present with small t, then the plant is dwarf, or you can say the recessive plant is observed. So dominant characteristic, the characteristics which are prominent and expressed in the successive generations are called dominant characteristics. They are expressed in the heterozygous form as well. For example, in a cross between a pure tall plant, that is capital T, and a pure dwarf plant, that is small t, what we observe, the first generation is capital T and small t, and all plants are tall. If we talk about the recessive characteristics, these are characteristics which are suppressed or not expressed in the successive generations. They are expressed only in homozygous condition. So when we cross capital T with small t and small t, the first generation what we got is the all tall plants, and the recessive character is hidden. The important term is monohybrid cross and dihybrid cross. The word mono means you can say one or single, and di means two. So the monohybrid cross when we are considering only one pair of contrasting characters, and dihybrid cross when we are involving with or carrying two contrasting characters simultaneously, or you can say together. So monohybrid cross are the cross involving one pair of contrasting characters and parents, referring to monohybrid cross. And the hybrid thus formed is monohybrid cross, like the cross between two plants with the, like if we are considering only the height of the plant. Similarly, if we are considering only the color of the flower, or we are considering only the color of the seed. So these are only single characteristics. So these are monohybrid cross. But if we are considering two characters simultaneously, like we are taking height, like height of a plant, as well as the color of the flower, like in the case of capital T, capital R, capital R. So we are taking two characters simultaneously. Okay, or we are taking the height of a plant or the seed color. So there again we are taking two characteristics at a time. Okay, the next or last one is your F1 generation and F2 generation. So first PL generation is considered as F1 generation, where the first generation of offspring obtained from the cross between parents. Okay, if we cross two parents, then the generation what we obtain is known as first generation, first filial generation. And when we cross between these F1 parents or F1 individuals, the interbreed, whatever we obtained in the next generation, are considered as F2 generation or second filial generation. So I think that's all about the terminologies in genetics. Okay, thank you.